Hello? Okay, I'm going to assume that it's recording. All right. Welcome back to another modeling session with Jennifer Lynn. Today we will be continuing my original model of Braftovi plus Mektovi, a combination treatment inhibiting the BRAF protein and MEK protein of the MAPK pathway. You understood those words? You're already doing great. All right, just going to create a new Safari file. Oh, I forgot I was supposed to screen share. How do you screen share though? Oh, there we go. Share screen. Of course, I'm going to share my Safari page. Silly Zoom. What else do you think I would share? Okay. All right, let's continue Braftovi and Mektovi. It'll be fun to talk about it. And it's in Pfizer, I'm pretty sure. There we go. Braftovi and Mektovi. Oh, I remember why I left this model for a while. I mean, there were some other reasons. If you look at its last edit, it was 12 days ago. One of the reasons I left it was because it had a complicated royalty structure though, and I didn't want to bother. But let's get to it. We have to deal with it. Pfizer can negotiate their royalty structure. I can model it. Come on. Okay, and this was a new drug, I believe. Okay. All right. We understand. And 2031, Mektovi expires. 2033, Braftovi expires. All right. All right, add a couple columns here. Okay. Seems like I have everything. I wonder why I was putting this off. Maybe we could do a fun lesson tonight where we finish this model and then maybe we'll go look at some science papers and news and we'll dissect it and look for market opportunities, and we could go through my thought process a little bit when we're looking for new drugs. I think that'll be fun. Yeah, I think I'm basically done with this. I just need to model out the rest. All right, scrolling to 30, 464, 696. Let's look at the other BRAF/MEK inhibitors. Braftovi+Mektovi, that one's an older drug, also a BRAF/MEK inhibitor. And let's see where the revenue peaked, because they're both just approved in V600E mutated melanoma. Okay. And Braftovi+Mektovi, they're peaking around 1.5, 1.7 billion. That seems like too much market share for Braftovi+Mektovi and not enough differentiation to prove it. But what do I know? And this is also US only. So Braftovi+Mektovi peaked in the US at around 600 mil. So what this is really telling us is that Braftovi+Mektovi might not make it by this much. We should be a little bit more conservative. Then let's do what's in here. It would be helpful if we also looked at why Braftovi+Mektovi are better than Braftovi+Mektovi. Maybe we could look at a comparison trial. Also, if my modeling is a little bit slow today, just know my mouse died. My mice died? My mouse died. So I'm not using it. Let's just drag this bad boy out to 3%. I really don't see this making that much money. Okay, and then Braftovi EU patent expiry. So the EU, when patent expiry happens, it's around minus 40%. Actually, let's do minus 30. Here, minus 50. I'll change this later, maybe. And then in the US, when patent expiry happens, there's a much steeper drop. Oh, this is annoying. I didn't separate the US and rest of the world revenue for the two drugs separately. That's bad. We gotta fix this. We gotta be meticulous. We gotta be rigorous. Oopsie daisies. Honestly, truthfully, I haven't done a model in like maybe 2 weeks. Yeah, 2 weeks. Straight up not having a good time. All right, let's fix this bitch. Okay, so both Brof and Mech are having EU patent expiries at the same time. That's good. And Mech's US patent expires 2031. Then you copy paste here. There should be this. Give me 1 second. Oh, this is fine. Yeah. For this new drug, I think the growth is going to be basically the same rest of the world as well as in the US. Let's see what it did before. Oh. Oh, oh my bad. I did not do this right. Okay. Let's find the way it was before. And the issue, which I didn't realize, is that we are only looking at Pfizer because the other two companies are private. And what we should have done here actually is Braftovi less growth, and then there we go. And the EU patent expiry doesn't actually matter that much since we're only looking at US for Pfizer. Okay. All right, so Braftovi US patent, actually. Whoopsie daisies. Okay. So then you will see minus 40. You'll see minus 50 here, actually. Minus 55, minus 40. That's average. That's fine. But Braftovi, US patent actually expires a little bit later. 2033. So you'll see minus 50 here. Minus 55, and then minus 40. That is the average pattern that you see in larger drugs once they go off patent. Okay. All right, and let's do COGS. SG&A. Let's just take Xtandi. Each company does have different cost of goods sold and sales, general and administrative costs, as well as research and development costs. So just look at the past 4 years, look at their 10Ks and get an average and you'll be fine. For Pfizer, oh, and taxes as well. So Pfizer doesn't pay that much in taxes. And if you're paying over 35% as a corporation, you're doing something wrong. You're a dumb bitch. Ooh, what happened here? What did I do? What did my dumbass do? Oh, okay. Let's change AE1 to this. Let's change that this way. Seems right. That all looks good. Except this part where it should still be A3. So yeah, the formulas are all right. Yep. Let's take that all the way down to here. And of course, as your patent starts to expire, you're no longer doing research and development on this drug. So R&D is at 5%. Oh, that's really low. Why is it at 5? Oh, because this drug already expired, or it's very close to expiring. Okay, yeah, no, normally R&D is at 10. So let's just keep that for a little bit. The cost of goods sold for a small molecule is 9%. Energy is at 10, 10, 10. As it gets closer to an expiry and you stop seeing trials going on, you are going to get 8, 7, 5. 5's good. 3, 1. Okay. Time to do some research and look into why this is a next generation drug that seems or should be more effective than Tafinlar and Mekinist, which is a blockbuster drug making about 1.5 billion each year. If it's better than Tafinlar and Mekinist, then it should also make a little less, but blockbuster range. So let's figure out what makes it different. Okay, so it seems they've demonstrated that Braftovi is a better BRAF inhibitor than Selboraf, which is a slightly older BRAF inhibitor than Tafinlar. That's all fine and nice, but yeah, there are no scheduled head-to-head trials that directly compare these two drug combinations against each other. If there was a scheduled head-to-head trial, it'd be very interesting to look at. But and if there's not, it means that there's reasonable uncertainty that these drugs or that BRAF plus MEK is not significantly superior to Tafinlar plus MEK, because the pharmaceutical companies are not willing to plow hundreds of millions into a head-to-head trial to prove it is. So that's fine as well. But when you're truly confident that your drug is superior, you do the head-to-head trial because it convinces physicians to prescribe your drug instead of the older one. See, whoever physician this person is, said it is sort of challenging to really talk about objectively are the differences in efficacy because of the fact that they haven't been compared head to head. See, this is a physician opinion. However, there are some fundamental differences. But guess what? It'd be a lot easier to just prescribe the superior drug if you had that trial evidence, which you do not have because Pfizer isn't that confident that they will come out significantly superior, which is always fine, but that's why I'm not making it a blockbuster drug and giving it as high of a peak revenue as the standard of care, which is Tafinlar plus Mekinist. Actually, let's make that a no. No plan head. Different side effects. Okay, that's helpful. Interesting. Seems that when patients are resistant to this BRAF-MEK combination, they're still reactivating the same pathway. Oh, interesting. That means there's other protein targets available in this pathway that you can try to inhibit as well to treat patients that show resistance to BRAF plus MEK. Oh, would you look at that? They now have ERK inhibitors that are in clinical trials, started as single agents, but will be combined with ERK is downstream of MEK. So MEK signals ERK. What could help here? Actually, they've got all three proteins in the pathway now. BRAF, MEK, and ERK. Those are the three proteins. I wonder if there's something else that's kicking back resistance. All right. All right, so we achieved our goal of looking to see if there's differentiation, and so far there's no trial proven differentiation. Maybe there's some differences in side effects, but it seems like it's not. It's just different side effects, not different levels of side effects. So that's interesting. And this seems to be a more potent BRAF inhibitor. So, it's good. It's good. It's interesting. Seems like we're done with this model though. So we can delete this question. What is Tiffin-Lar priced at? Wow. Pricey. Seems like Braftovi plus MEK is cheaper than Tiffin-Lar. Ooh. Let's read this ASCO post just for fun. I think I'll be almost finished, and then we can walk through endpoints news, and look at what's popping in biotech lately. What's hot, what's not. Who's getting an investment from raw capital? Look, this presents oncologists with a unique dilemma. They're already approved to be RAF-MEK inhibitors, so why switch? Please answer this question for me, Mr. Dr. Scharfman. Similar efficacy, but different side effect profiles. Thank you, doctor. Difficulty. I'm just going to read the headlines. I don't have time for these paragraphs. Okay, just seems similar. Like, what are you supposed to do? Tiffin-Lar mechanism patent expiry in 2027. Braftovi+Mektovi revenue might drop in 2027 then. You know what? Let's put them together. Insert row. Insert row. This is going to affect revenue. These drugs are quite similar, so when they create biosimilar generic drugs of the mechanism to Finlard, patients may opt for the cheaper option if there is not too big of a difference. What did I even put here? This was to Finlar. Yep. Okay. All right, so it's at 1% revenue. It's going to drop a little bit more here. So you might even drop more. I don't want to be too nice to this drug. Okay, I just want to be conservative. Oh, fuck. I want to be conservative about our estimates here. Okay. This seems about right. And the peak revenue that you'll see here is the 700 million range, which is actually quite a lot. Maybe we should give it less, huh? This drug is approved for more than just melanoma. Yeah, no, fuck, no. Braftovi+Mektovi may be approved for things in the future, but the probability of success isn't 100%, so we should lower the revenue. Yeah, okay. Let's do, man. Let's do like 18% here. I'll make me more comfortable. Let's do, it's a weird number, but whatever. 35. Hmm. I don't see what's so great about this. I feel comfortable with these numbers. let's just, I think this will be fine. Okay. Oh, first quarter for Pfizer might have come out already, actually. Pretty sure it is out, actually. It's earnings season. So we could take a look at that. Should I include the Pfizer royalties from Braftovi+Mek? Because I know they received royalties. It's just a mystery of how much. 2828. That seems a little bit low. Oh, but it's only US revenue. Let's just include Pfizer royalties. Let's do royalties here. Insert row. %. Now let's just assume the rest of the world is very close to what Pfizer is making here in the US. Oh, no wonder. You add in royalties and you get $3.3 billion. That feels pretty good. I think I'm going to leave it. All right. Let's call this done. What we're going to do is we're going to end points now. We're going to have a little bit of fun reading the news, and I will probably do this for a couple minutes, and then we'll end early. Sorry that this hasn't been the most entertaining stream about my personal life, but hopefully I walked you through my thought process of completing Finlar Mechanist a little bit. All right, let's just look at all news. Astellos walks away from 450 million cytokinetics pact after taking a few extra months to see progress. Oh boy. Oh, but this isn't very cancer related. Okay. Zero, one. Oops. Okay. Oh, there we go. I offered Josh Belenker to join my startup. He said that he was busy starting his own thing, and he did. Did a good job. Very impressive. Seems like they're working on a new technology called molecular machines to target proteins that are constantly shifting. It's interesting. That's very cool. It's always good to be up to date. I think Josh Belenker's startup is called Treeline. Okay. Ooh. PD-L1 reckoning, that wasn't. There's about 7 or 8 PD-1 inhibitors in the market right now, and it's very competitive. The big ones are all making like almost 3 billion a year. Oh, this isn't that interesting. Biotech. Oh, it's a blood treatment. Blood cancer treatment. I see. Or is it multiple sclerosis? Oh, interesting. But CD20 is a blood cancer target for non-Hodgkin's lymphoma. That's also true. Ah, that's interesting. They're repurposing it, or they meant to develop it for cancer, but now they're taking it into multiple sclerosis. It sounds like they probably weren't. Interesting. GC4419. What? Superoxide dismutase mimetic. What? Oh my lord. Oh my gosh. Hmm. Interesting. I feel like this is some kind of oxidation mechanism. Could be interesting. I thought it was going to be more MAPK pathway because most of pancreatic cancer is caused by KRAS mutations. Actually, like 98%. So I was more interested in if it targeted KRAS or not. Price competition. Interesting. This is another PD-1 inhibitor, Opdivo. Law non-coding DNA. Oh, interesting. Mm, that's true. I wonder how they're targeting the extracellular DNA. The question is how they're going to target it. Okay, let's find out. Mm. All right. Yeah, seems like it's going to take a while for them to even announce a candidate. A couple years before they get into phase 1 trials. It's very interesting. Okay. All right. Let's read a little bit of BioRxiv. Defines. That's a big word. P53 deficiency is a big issue. Can we drug run X3-MYC? Let's see. Osteosarcoma, bone cancer is characterized by that. Many cancers are. However, the molecular mechanisms man, this is interesting. I think MOIC is hard to target as well, though. Run X3 knockdown. Are there current Run X3 inhibitors in development? It's hard to make transcription factor inhibitors, though. But not impossible. Well, let's look into it. Let's see. Let's look at the mice data. Okay, overall survival. It seems these survival curves are a little too close to each other. I'm more comfortable if they were further apart. It would mean there's a more significant difference in overall survival. That's interesting. TRPC1 is a predictor, and they're saying that when there's presence of upregulated or more TRPC1, the chemotherapy works better. But there's not much you can do about that because it's very hard to make a drug that creates more of a protein. It's much easier to inhibit. Look, we were just talking about BRAF inhibitors, Braftovi. And when it sees, when patients show resistance, which means when their tumors grow larger, there's usually some type of mutation happening. And this paper is talking about what exact mutations are happening. So it seems like if you're inhibiting this kinase, called ARCA, BRAF inhibitor resistant cells with A375, stop becoming resistant. Now, let's always be critical of papers. So you can see about 2 questions off the bat, which is vemurafenib is a very old BRAF inhibitor. It might even be off patent right now. So it's not the best, it's not, you want to use the newest inhibitor to show resistance. You want resistance to be seen in the newest BRAF inhibitor so you can fix it. Number 2, it's not being combined with an MEK inhibitor, so there's a very good chance that this resistant cancer, if you add in an MEK inhibitor, then your ARCA inhibition no longer matters because it might just overcome the resistance better than inhibiting this ARCA kinase. So those are 2 problems that you can run into, and it doesn't seem too significant. Okay, I'm just gonna skim through these now. Oh, Atross. Atross is a target I'm very interested in. Pretty sure there is an inhibitor. There's a startup working on a V1. Okay, DB01. And they're already in trials for Ross cancer? Probably not, but that could be a good idea for them. Okay, well, whatever. The idea is that someone already made an inhibitor. ERB2 is a protein that is mutated in prostate cancer sometimes and can also be targeted there. Okay, it seems like if your cancer is still growing after ERB2 inhibition, there's 2 transcription factors that could be contributing. Not too significant because it's hard to target transcription factors. It causes a lot of adverse events. And they're also, these proteins move and change shape a lot. So it's hard to get an inhibitor that's really potent and attracted to a specific site. Neuroblastomas, kind of rare. Okay, let's go on. The amplification. Oh, there we go. ARCA comes up again. That could be interesting. Let's take a look. Last paper before we end. I just want to look at the mice data really quickly. Mice, 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 mice. We love our mice. This is all binding data. I want to see how it binds. I understand it binds to the ARCA protein. Show me the mice. Ah! This is all in vitro. Oh, I guess there is no mice data. Huh. Well, let's just look at results really fast. Not materials and methods. There. Okay. That's cool. Hold up. This could be not bad. This could be not bad. Hmm. Neuroblastoma is rare, but so what? With amplified MYCN. Gosh, I feel like this video is so like just so random. MYCN amplification in 20 to 30% of neuroblastoma patients, and overall survival is still less than 50%. Okay. So basically what the paper is saying is if you're targeting, you have an inhibitor, there are inhibitors made of this, for this Aurora A kinase inhibitor, and what Aurora A kinase does is it's supposed to take MYCN to the proteolytic trash can, which is a protein that will just destroy your MYCN. And actually, no, it's supposed to prevent MYCN from being degraded. So now if you inhibit Aurora A, it prevents Aurora A from interacting with that MYCN. And therefore, MYCN is now able to be trashed in your protein trash can. That's not bad. That's not bad. And there are Aurora A inhibitors out there, but it seems like they might not be potent enough and may need more research and development so that they can become a good deliverable drug. You know, I think I'll look into it. We'll see. Nature papers are always good, and yeah, I'll look into it. Cool. We have something interesting that we've discovered tonight, so we should be proud of ourselves. I don't know what's happening on the screen right now and whether you're just seeing me or not. Oh my gosh, you're seeing me. But yeah, you know, this was interesting. I hope that you learned a little bit more about BRAF and MEK inhibitors, and that you were able to learn about potential drugs in the cancer space that could take some development, and also some new companies coming out that are working on new drugs, new therapies. It's a very exciting field. It's very fun to be in. All right, it's almost my bedtime, so I'm going to go to sleep on my comfy bed and wake up early in the morning. And I hope you guys can do the same tonight. So have a good night.